Hello everyone, my name is Jiayu Shang. In this presentation, I will show you our recent work about phage protein classification. First, I will briefly introduce what is phage and why we need to study phage. Phages are the most common and diverse entities in the biosphere. They are danger killers in the world and kill trillions a day, but fortunately, they mainly kill bacteria. During the life cycle of the phages, they will infect the bacteria and replicate within them. Then they will kill the bacteria to release their offspring. Several phage applications have been used in our daily life based on their properties. One of the most famous one is the phage therapy. In phage therapy, phages can be used as the superweapon to treat bacterial infection. In addition, compared to the antibiotic, phages are targeted. They will only kill the bad guys in our body without harming healthy cells. Over the past decades, a large number of new phages has been sequenced using metagenomic data, which can sequence all genetic materials in the environmental samples. Given the metagenomic dataset, there are two fundamental goals for phage analysis: identifying phages and predicting their hosts. A common challenge for this task is that many proteins in phage genomes hasn't been annotated. So with very limited protein annotations, it becomes challenging to distinguish phages from other microbiomes and link phages to their bacterial hosts. So in this work, we aim to classify phage proteins to advance the downstream analysis. In the following slides, I will show you the problem formulation and our model design. Then I will give you some examples of how the model can assist the analyst in phage-related tasks. Traditional methods for phage protein annotation are based on the exact match between sequences. However, recent research has shown that the number of the known phages is just the tip of the iceberg. In addition, phages are diverse. It's estimated that about 67% phages proteins don't have alignment results with the annotated proteins. The alignment-based method usually lead to a low recall performance, so computational methods for phage annotation is in great demand. The protein of the phages can be roughly divided into two groups: virion protein, PVP, and a non-virion protein, non-PVP. The PVP refers to the coat of the proteins, and the protein in the non-PVP are usually different kinds of enzymes. In the design of the computational method, protein annotation is the classification task. The input of the model will be protein sequences, and the output will be the function or label of the proteins. Phage protein annotation is a well-defined problem, including two successive tasks. The first one is the binary classifier that will classify the protein sequence into either non-PVP or PVP. Then the PVP proteins will be predicted into a more specific task, such as tail, head, or fiber. People care more about the annotation of the PVP because they are usually used in the phage relative task, like phage identification or taxonomy classification. In our model, there are two main components: K-gaming representation for encoding the protein sequences, and the Vision transformer for classification. Because the proteins are string type contains 20 alphabets, traditional encoding methods such as one-hot encoding will lead to a sparse matrix which is not suitable for machine learning model to learn. In our work, we employed K-gaming representation to encode the protein sequence into images. Different protein will be converted into images with unique patterns. The idea of CGR is from chaos gaming theory to construct fractals based on the input. In the original chaos game, it has 3 different colors. In each step, we will roll a color and take a move. Here is an example of the moves: red, green, blue, and blue. In each step, we will move forward towards the corresponding color. So intuitively, it can be easily to extend to DNA sequence by adding one more direction. Let me give you a more detailed explanation of how we convert the DNA sequence into the images. Here is the recursive function, and as you can see, the next move only depends on the current position and the direction. The current position will be pi-1 and the direction will be Vij. For example, if we have a sequence CATG and imagine we will start from the 0, 0. The first letter is C and corresponding to the row, both Vi1 and Vi0 will be -1. So we will take one step from 0, 0 to -0.5, 0.5. And we continue the move according to the rules and finally form a unique path. Here are some examples of encoding the whole genome using the CGR. And as you can see, compared to the random sequence, it's clear that CGR can encode the genome with useful information, and different genome images contain different patterns. So the CGR images are suitable for the pattern recognition method. However, if we want to use the CGR image to train a model, there is still a problem we need to tackle. Because the length of the genomes can vary a lot, the resolution of CGR will be also different. Let's use the simple example to gain more intuitive understanding of this issue. Back to the sequence CATG, when we record the precision of each step, we need to compute the x-axis and the y-axis in the coordinates. Although in human, these coordinates are only 2 float values. In computer vision, the value of the image is recorded by a matrix where both index of the row and column are integers. So we still need to convert the float coordinates into the integers. And with the increase in the length, the image will become larger and larger, resulting in image with different shape. But most of the models can only handle image with the same shape. So what we need to do is to scale the CGR images into the same resolution. Scaling the images are common pre-processing progress in the computer vision field. Here is an example of scaling the Mona Lisa figure. Although these 2 figures have the same shape in human eyes, the high-resolution one was represented by a much larger matrix in computer. Although this low-resolution figure is not that clear, you can still recognize it is Mona Lisa, so the resolution will not affect the patterns a lot. In CGR, we call it a frequency matrix. The scaling process will first divide the figures into many grids and sum up the pixels in the grids. Then, the original images can be represented in a low-resolution image. We will apply the same scaling methods on all the genomes so that the frequency CGR images can have the same shape. Once we have these images, we can apply the models in the computer vision field to capture the patterns and learn to classify the images. One of the most famous models is the vision transformer. It is a variant of transformer that aims to learn the contextual information from image data. The large language models such as ChatGPT has demonstrated their ability in our daily life. All these models are based on the transformer model to learn the contextual information from the language. Here is an example. The transformer will capture the features such as importance of the words and the association between words for prediction. Because of the powerful usage of the transformer-based model, researchers also try to adapt it to the image processing field. When handling image inputs, we will split the image into several patches so that each patch can be treated as a word. Then this model can learn the importance of the patches and the association between patches. The process of learning can be roughly divided into two parts: the patch embedding and the self-attention block. These two processes can be viewed as learning local and global information independently. In the first part, the embedding are learned from each patch, and in the second part, the self-attention block will learn the correlation between patches. This attention score represents the importance of every patch pair, and both information will be used for training and prediction. So based on the introduced techniques, we design a model named PhaVIP for Phage VIrion Protein classification. First, we will encode the protein sequence into FCGR images. Here is an example showing the images of different virion proteins, and it is clear that there exist some unique patterns in different protein images. Then we will generate patches from the images and feed these patches into the vision transformer model. In the prediction, we train two different classifiers. Only those proteins predicted as PVP will call the multi-class classifier for further classification. Next, I will show you some experimental results of our PhaVIP model. In the experiment, we use standard classification matrix for evaluation. We benchmarked PhaVIP with the latest published tools on several datasets and showed two applications of the PhaVIP advanced the downstream phage-related tasks, including host prediction and taxonomy classification. In the binary classification task, we have two methods to split the dataset. The first one is split by time, which is more related to the real scenario. All the phages released before December 2020 will be used as training data, and the remaining phages will be used for testing. The results shows that PhaVIP and DPVP can both achieve good performance. And in the second data construction, we create a harder case. We split the data by sequence similarity. We will first estimate the similarity between the protein sequence in each annotation, and then we will set a threshold to control the similarity between training and test set. In this experiment, as you can see, it's expected that the performance becomes better with the increase of the similarity. PhaVIP always has the best performance across different similarity thresholds. We also apply the same data construction in the multi-class classification task. The gap between PhaVIP and other methods shows that PhaVIP can have a more stable performance in the multi-class task. If you are interested in our evaluation design, feel free to explore the additional experiments we conduct in our paper. Next, I will show you some application of PhaVIP. The first one is the taxonomy classification for novel phages. In this experiment, the dataset contains phages in the RefSeq, and the aims of the task is to assign a family taxa to the novel phages. We use vContact, which is a widely used tool for phage taxonomy classification, as a baseline tool. In the original design of the vContact, it will use all the proteins from the phage sequence to form a protein sharing network and do clustering algorithm on the network for classification. In our experiment, we designed two variant versions of V-contact. Only use PVP version and only use non-PVP version. We'd like to check whether these two variants will affect the performance of phage taxonomy classification. Here are the results. We record both accuracy and the running time of V-contact. We also randomly sampled the same size of proteins as PVP and non-PVP version as controls. As shown in the experiments, PVP always has a higher accuracy than the non-PVP version. And although the accuracy of both variants will not affect a lot, the running time decreased sharply because of the size of the database. Thus, in the future design of the taxonomy classification, PVP should draw more attention than the non-PVP. In the second application, the host prediction task, we will show interesting findings that may contradict our consensus. The dataset used in the host prediction contains the phage bacteria pairs. Each pair represents a real infection, and the aim of the task is to predict the host for the input phages. In our design, we will use the basic alignment method plus P for the host prediction. And as you can see, we create two different databases, including the phage protein database and the bacterial protein database. We will estimate the similarity between the test phages and the database for prediction. Also, in both cases, we can either apply only PVP search and only non-PVP search. For each test phage, we will use majority vote on all the aligned proteins to give a prediction of the host. Intuitively, because PVPs are more specific, researchers usually use them to design host prediction models. However, in our results, using non-PVP for prediction always has a better performance than using the PVP, especially at a lower rank like species level. We try to analyze the reasons and find out that some enzymes are more specific for distinguish the host. So maybe non-PVP can be used as an indicator for the host prediction task in the future. To draw a conclusion, we employ the CGR to encode proteins into images and incorporate the vision transformer to learn the unique features from the protein images. Our experiment not only show PhaVIP has a good performance on different datasets, but also demonstrate the usage in the downstream phage-related task. In the future, we'd like to extend the PhaVIP to the multi-class classification on non-PVP data because of the potential needs. Also, our methods are general solutions and can be adapted to other data or problems not related to phage proteins. Here is the GitHub of PhaVIP, and we also provide a web server Phagebox for the phage sequence analyst. In Phagebox, you can run all the phage-related tasks, including phage identification, taxonomy classification, lifestyle prediction, and host prediction. All the methods behind are designed by our group and proven to be the state-of-the-art solutions. We also provide a visualization of the results for reference. Feel free to have a try. Thanks for your listening.